So we're having Jonathan with us here. Jonathan is my companion and he will be paying attention to what I'm doing, as well as the rest of you. So, Jonathan, we're having here, we're going to start here, students, with a Petri dish again. Let's say this is our Petri dish. This is our animal cell. And the animal cell does not have a cell wall. Is that clear? So we're having small vacuoles. And the reason why animal cells have small vacuole is because they do not need to store a lot of food. They can go and get food. Plant cells, however, have a large vacuole because they need to store for consumers. So, this is our animal cell. This is our vacuole. This is water. So this container is filled with water. So there's a lot of water here and there is less water in the cell. So what will happen in this case is that water molecules will move from the Petri dish, which is outside of the cell, into the cell because it is high in concentration out of the cell. It will move into the cell as well, and then it will cause this cell to become bloated. The vacuole will get large. So where or how water is moving? It is moving through the cell membrane, then it is moving into the cytoplasm, and then it is going into the vacuole. When that happens, let's say you leave it for 15 minutes or 10 minutes, then this is what you will be seeing. You will see a very bloated up cell, a very big cell, turgid cell. It will get large as well. But if water, but remember water will keep moving into the cell. As water keep on moving into the cell, then obviously after a period of time, the cell will burst. Why the cell will burst? Because there is no cell wall. There is no cell wall. So the cell wall in the plant cell will protect it from bursting, but the animal cell, it will become turgid, and if it keeps on taking in more water, it will burst eventually. Do you understand that, students? Is there any question? So it's the same thing we're going through, but the only difference is the cell wall here, and this is not protecting it. This is going to cause it to burst. This is also called hypotonic, because water is moving into. This is also called turgidity or rigid. All right. Now, in a cell such as your red blood cell, your white blood cell, or so on, all of this can happen. Let me just pull up this here for you to show you a diagram. Look here. So here we have our red blood cell. Everybody could see the red blood cell here. So we have our red blood cell. And if this red blood cell is placed in distilled water, this is what will happen. Do you see how big it becomes? After it becomes so big, then it will swell and then it will burst. Why will it swell and burst? Because there is no cell wall around it. Get that there, students? In this case, it's called It's called hypotonic. Good. So that's an example of what happens with an animal cell. It can burst very quickly. Now, I want to bring this to your attention. I know of a very severe case where there were parents who actually forced a child to drink 1 bucket of water, and the child literally drowned. When the child does not mean the child went in water and drowned or pushed the head in the water and drowned. The child consumed over the amount of water, the extra amount of water, and causes the cells in the brain to burst. This here can happen if you were to take too much of water and any stuff, any concentrated material in your body. This can happen. Okay, so be very careful. If you drink too much or excessive, remember I'm telling you about a bucket. Okay, all right, so let's move back. Yes, Jonathan, tell me what you understand about this animal cell. What will happen to this animal cell here? Quickly.
Miss, I understand that when we say the water in the Petri dish, which is outside of the cell, right? The animal cell will move to the animal cell, right? And then it will go to the cell membrane, which will then go to the cytoplasm, then to the vacuole, right? And then when the water moves there, it causes the cell to be more bloated or large, right? which end up also being turgid. When the water move into the cell, but it will move to the cell because the concentration outside is higher than the concentration inside the cell, right? And if there is too much water inside the cell, it would end up bursting because there is no cell wall to protect it, right? And this is also known as hypotonic.
Very good.
And yeah, it is basically
So, which cell will burst faster if you're going to expose it to a lot of water? A plant cell or an animal cell? Both is left for...
Miss the animal cell.
Alright, wonderful. Is there anything else anybody would like to add or ask before we move on to the next cell content quickly? So everybody understand hypo? Yes. Quickly?
Yes, miss. Move on.
We could move on?
Oh, well done.
Okay. So here we're moving on to another cell situation which is called hypertonic. Now, let's see what happens. This is the opposite of hypo. So again, we have our petri dish, we have our plant cell, we have our vacuole. Now in this case, let's say we don't have water, but we have something that is very highly concentrated or not so dilute. So we have here sugar solution. So sugar is glucose, so it's C6H12O6, glucose. Alright, so here we are having sugar solution, and this sugar is having a lot of water or less water. Which one is it? So think about, we can have sugar or we could have salt, any one of them. But this solution is very, very concentrated, meaning it got more substance, it got more density. The question I want to ask is, where is the water? Where there is high concentration of water here? In the cell or out of the cell? Where will it be high in concentration, water concentration or water particles concentration? In the cell or out of the cell? Now it's very simple.
Inside of the cell.
Inside, very good. Now what will happen in this case is that because this is sugar solution, we know sugar solution is rich. So water will be in here, it is high here, and it is low here. Now think about, if you were to take some potato and leave it in sugar solution, what will happen over a period of time? Or take some pine and put salt in it, what will happen afterwards? You will notice that there are going to be the pine or anything that is exposed with sugar or salt, it becomes very floppy. Okay, so what is basically happening is that the water molecules are going to move out rather than in, because the water is inside of the cell than outside. So it's high in the cell and it's moving out. So once outside is very concentrated, the water is going to move out. So water molecules are highly concentrated in the cell and it's moving into the concentrated solution, which is the sugar solution. So after a period of time, this is what you will be seeing. Let's say 5-10 minutes. The cell membrane will pull against the cell wall and everything will look very squishy. And the cell will become very small. In this case, what do we call here? Because everything is dried out. It's like you going into the sun. So water is leaving your cells and it's coming out. Or solvent particles. If you go in salt water, the same thing will happen to you. Okay, water is moving from, into, or out of your body. Alright? So this is what is happening. Osmosis is present in every single thing, but which cell content or what type of it is happening? That is the most important question. So in this case here, it's going to be very, what should I say? It is very stringy and it is very flaccid. Very good. So it is flaccid and it shrinks. So this is how the cell will look. The parts of the cell will pull against the membrane. It will lose its water content and it will not be able to perform its activities because everything is drawn out there and the cell can die. Okay, students. So in a season or in the day where the plants them look very withery and dry, this is what is happening because it's losing a lot of water. And when it's raining, it looks fresh and turgid and erected. It is gaining water. So do you understand which osmosis is occurring? What type of content is present? So in an animal cell now, what will happen in this case? We will have the water again. We have salt again here, or sugar. Let's say we have sugar. What will happen in this case? The same thing will happen. It will leave the cell and come out. It will look very shrinky and small. All right? And the cell will eventually die. In this case, it's also called flaccid, or it becomes shrink. All right? Good. So that is a case of flaccidity, students. So we can also define this state as plasmolyzed, that the cell is plasmolyzed. Okay? Now, one person will explain this to me, but before I ask a person, I want to make this point clear, that we don't need to have concentrated solution here. We can just leave the cell in a container with nothing, and you will see that there is no water here, so water is in here. So water is moving out to come where there is no water. Is that clear? So if you leave a piece of meat and you go back after 5 minutes, you'll notice that the solution is inside that container there. How did that happen? You did not put anything there. That is because the water particles are moving where there is high inside the cell to the lower concentration, which is outside of the cell. Understand that now? So that is how it occurs. One is into, one is out. Hypo is into the cell, hyper is out of the cell. Okay? Yes. One person will explain to me what happens in a hypertonic situation in a plant and an animal cell. One person quickly. Volunteer? Or I will call your companion. Yes.
Miss in the animal cell or the plant
Both cells, my dear.
Both?
Yep.
Alright. Miss in the plant cell. First things first is hypertonic, which means the concentration would be higher inside of the cell, right?
The water concentration. Stop. The water concentration is higher inside of the cell. Okay?
Yeah, miss. The water concentration. Yeah, miss. And the concentration of water is lower outside of the cell.
Good.
Right?
So that means that we don't need to have water outside of the cell. We don't need to have anything outside of the cell. But we can also have sugar or very rich solution. Okay?
That would absorb EMS.
Good. Continue.
So now the water will move from the vacuole, go to the cytoplasm. It's like the whole reverse. Go to the cytoplasm. Then through the cell membrane and out of the cell into the petri dish or to the other solution that would absorb the water, right? And and basically the cell would leave flaccid, right? Whereby as in the animal cell now, the same process would happen, right? But because it doesn't have a cell wall, it would show more flaccidity, right?
Yes, very good.
Yeah, miss. Which basically that is how hypertonic does work.
Very good. So he mentioned a very important point that I did not mention to you, that in the plant cell there is a wall around it, so it doesn't look that, stringy at the end of it, because it still carries a structure, right? That's what the cell wall is for. But for the animal cell, there is no cell wall, so basically what you will be seeing is a structure looking like this. Let's just go back quickly to our slide here. So you see what happens here is if you put it in water, you'll get this. And here, if it's placed in concentrated salt solution, see that? If it's placed in concentrated salt solution, then water will leave the cell, and you see what happened here? It becomes shrink and it shrivel. Everybody get it there? So it becomes shrink and shrivel. In this case, we say it is hyper. You know, somebody irritate you and you get hyper. What's happening there? You are releasing a lot of heat. So if you're hyper, you're releasing a lot of heat. And if you are hypo, then you're looking all fresh. Right. Good. So I want you to remember that. But the most important point, students, is to remember where water is highly concentrated. And there's where it will move. Is that clear? Good. So in this case, water is in here. The petri dish will move into the cell. And this case, there is no water, but it is going to move out of the cell. So the next case of cell content is called isotonic. Isotonic is our last cell content, so pay attention. Now, iso means the same. Something say that you're iso. It says that you are same. The same extracellular fluid that has the same osmolarity as the cell. So, extracellular means it is outside. So, we're drawing our two types of cell. So, we are drawing our petri dish. And here is our plant cell. And our vacuole. And here we're having a fluid that is isotonic. So, what do you think isotonic means? It means to say that the same concentration of the fluid that is inside of the cell is the same concentration of fluid that is outside of the cell. So it will be let's say this is 3 in concentration. Let's just put a number. There's also 3 in concentration here. It's the same substance, the same density, the same concentration. That means to say who will move to go where? Okay? Who will move to go where, students? The same amount that moves into will be the same amount that will move out. So basically what happened is that there is no change. There is no change in the shape or the structure of the cell because nobody is based. There is no net movement. There is no net difference of it. Because the same concentration is here inside and the same is the outside. So it is said to be isotonic. Now, hypo and hyper is where there is a difference in and out. But iso is when they are the same. Understand, students? So the tissue or the plant tissue here or the plant cell is of the same size and consistency. And the reason why is because the solution is of the same concentration. Now, in this case where we have an animal cell now, we'll also draw here animal cell. Let me use the red for animal cell quickly. So we're having the animal cell here. Let's say we have our red blood cell and we have our vacuole. And let's say this is 3 and this is 3 in concentration. What will happen is that neither will it move in and out. Would there be a difference? So the cell will remain the same. Now, in other words, how would your cell look? How would you describe your cell in an isotonic situation? The cell will be not flaccid, but it will look very bloated as well, normal. It will not shrink because it's not losing. But it's not gaining so much to be so bloated. So it will get a turgid shape. Everybody get that there? It's gonna get a turgid shape. Turgid shape. Now in isotonic situation, they will not burst because it's not overtaking anything. Everybody get that there? So isotonic situation is there. hypotonic, and hypertonic. These are the three cell contents that are present when we're talking about osmolarity. Yes. One person to go through this for me quickly, and let's move on to our conclusion. Yes. Please volunteer quickly.
Yeah, Miss. For
Go
the
ahead.
isotonic. Water will need to move in or out, right? Basically, it will just be in one state all the time, right? It would look slightly turgid or bloated, and it would have a fixed state, right? And it will not burst because too much water will not be going in. So it would be in one fixed state all the time, and it will be the same. It will not be too big or too small, and just be slightly turgid, but not too turgid.
Now, you made a mistake there and say something I don't like. I want you to use the word same in concentration. Okay?
Same in concentration.
Yes.
Okay, Miss. Yeah.
Same in concentration. Because it is a concentration that allows things to move in and out. Okay? So if it is same in concentration, that means to say it's neither moving in nor out. It's creating a balance. Go ahead, do the animal
Miss,
cell
thanks.
now.
In the animal cell, it would be basically the same thing. So yeah, it would be the same. Yeah, alright. It would be basically the same thing. The concentration would be at a fixed point. It would keep the concentration would be the same both in and outside of the cell. So it would stay at one fixed point, and it would not burst because no water is there to overthrow or make it more big.
Very good. Wonderful.
More
Well done.
more bloated, sir.
Wonderful. So we would have highlighted the three different types of osmolarity or cell content or tonicity. Now those are hypo, hyper, and isotonic. Now let's do a little quick quiz here or recap of what we would have studied today. This is a diagram to show you how it looks. So this is your cell. And as you can see, when it is hypertonic, the water molecules are leaving the cell. It's leaving the vacuole. Do you see how the vacuole here looks? It's shrinking. So it's leaving the cell. It's coming out. When it is hypo, it is going into, and the vacuole becomes bloated. And the cell eventually becomes bloated. So we say that it is turgid. We say that it is turgid. If it is coming out, then it is plasmolyzed. And then if it is balanced, there's the same concentration in and out, then it is going to be flaccid. You can also use the word flaccid because it's neither turgid nor plasmolyzed. All right, so this is a nice diagram for you to remember. This is a case in terms of a red blood cell or RBC, of what will happen, how it will look if the cell shrinks, water is leaving. If it is bloated, water is entering. Or this one here is becoming very large, and here is net flow in and out. Basically, there is no difference in your net movement. So I have a quick quiz for you before we go. Yes, and I would like to have 3 volunteers to tell me what cell content is present in A, B, and C in the 3 different solutions. Quickly, I would like to call 3 names. So I'm going to call on Samira Juman. Samira Juman, you're with me. Tell me, the first cell content should be A. The first cell content will be described as what situation? Samira, you could hear me? All right, let's have somebody else quickly, a volunteer. 3 persons. A is what?
Mesa hypo.
This is hypertonic.
A is, go slow for me.
A
A is a hypertonic solution, mainly
Could you
because
please
the water
spell
is
that for
leaving
me,
the
because I
cell,
can't
so
figure out if you're saying hypo or hyper. So I want you to be
HYPOTONIC.
HYPO. A is HYPO.
Yes, miss.
Are you sure?
This is hypertonic.
Sorry, hyper.
Come on. So A is hyper.
Hyper.
Why is it hyper? Why is it hyper?
Means because the water is leaving the vacuole in the cell and going to somewhere exiting the cell.
Where is
somewhere that
water
has a
highly
low
concentrated? Stop. Where is water highly
the vacuum.
concentrated? In the cell or out of the cell?
inside of the cell.
Very good. So it's moving out. Good.
Outwards.
Great. That's correct. Bravo. Let's move to the second one. B. Another person.
B. Somebody else.
B is isotonic, miss.
B is
Isotonic.
isotonic. Why do you say isotonic?
Because water outside and the water inside is equal. There's
In,
a net
equal in.
concentration.
Good. Now I don't want you to tell me about water. I want you to say water particles. Okay? Good. Correct.
Yes.
Wonderful. And the third one, solution 3, is, let's have a female. Yes, female. One female there.
It goes into the cell, causing it. So outside is more concentrated, and the water goes into the cell, causing it to bloat and such.
Mm-hmm. So what do we call the cell content?
Hypotonic.
Very good. Wonderful,
Turgid.
students. It becomes hypo or turgid. So just a quick recap for our session today. I'm going to ask persons randomly. I'm not going to call names, so you will just volunteer and tell me what did we learn or what is osmosis now. Tell me quickly, what is osmosis?
Osmosis. Is I can go?
Yes, go ahead.
Osmosis is the movement of water particles from a region of high concentration to a region of low concentration through a semipermeable membrane until the concentration is equalized.
Wonderful.
Beautiful.
Good. What are the 3 types of cell content?
Miss, I will take this one. Or you want to go?
Go ahead. After that, you're not answering any other question.
Okay, miss. The 3 types of cell content are hypotonic, hypertonic, and isotonic.
Very good.
Explain them.
No, that's all right. I would like another person to tell me what they learned about hypo and where can we see hypotonic. Quickly. What is hypo and where we can see hypotonic situation. Yes, one volunteer quickly. Let's call on Shanika boys quickly. Yes, Shanika boys.
Miss, can you please repeat the question?
Tell me about the hypotonic cell content and where we can see that.
Miss, hypotonic. In a hypotonic, the cell is turgid. Yes, the hypo means less than.
Where is water high in concentration and where would it move to?
Miss, water is higher concentration outside the cell, it moves inside.
Good,
the cell.
wonderful. Now tell me of a situation where we can see a hypotonic cell content in life, in reality.
Miss, using potato cups.
Okay. So you do potato cups at home in your kitchen?
Yes, miss.
Okay, what you do with the potato cup?
Miss, stuff it.
Oh, you do? Oh, okay. My bad. Sorry. Wonderful. Thank you, Shanika boys. Let's have another person telling us about hypertonic now, quickly, and where they could see hypertonic. Yes, a female, please volunteer yourself quickly or I will call on you. Let's have Caitlin. Yes, Caitlin.
Miss Caitlin is not here.
Yes, Caitlin, go ahead.
Esther.
Okay, what is your name? Please state your name.
Esther.
All right, go ahead.
Miss, can you hear me?
Yes, I could hear you loud and clear. Go ahead.
Okay, Miss. Well, an example would be like if you soak vegetables, especially like the celery, for instance, when it's dripping water which is outside, it's higher in concentration, will go into the cell, which is in lower concentration.
I'm asking you for a situation of hypertonic, not hypotonic. So I want you to give me an example of a hypertonic situation. Okay? Remember, hyper is different from hypo. So I want you to answer that question for me. Re-answer that question quickly.
Miss, I could answer.
All right. Let's see if Madam needs some help there. Madam, do you need help? Madam is not answering. Go ahead. I have
Yes,
seen that the
Johnson
male
again.
is dominating the floor tonight. Go ahead.
Yes, Miss. Miss, an example of hypertonic is, let me give you an example. Somebody that goes and swim in a swimming pool. You know, after a while when you're in the swimming pool, your skin just starts shriveling up. Mainly, it is because, just how the hypertonic is happening, waters leave the cell and go to a higher, a place of higher water solution, which would be outside the cell. When swimming in the pool now, it leaving your body and going into the water around you. And that's why your skin just leaves shrivel up. And this is an example I got.
Okay, wonderful. Great.
Here.
The last one now is
No.
isotonic. One last one is isotonic. Yes, Corwin, go ahead.
Mr. Nice.
Good night.
Okay. An isotonic solution will be oil in water. The oil will not mix with the milk.
Oil in water.
Yes, miss.
Why would you want to say oil in water? Because remember, we're talking about no net movement, right? There is a little, but there is no difference. Okay? So I want you to come with another example to me, dear.
Yes, I can.
All right. Let's have somebody else helping you out
Only thing I'm
there.
thinking of right now, miss, is beer spelling.
Okay. Let's have somebody else helping you out. A female quickly. Costello, you want to give a try with isotonic?
Miss, I would like to
Miss
ask.
Destiny is not here neither.
Yes, Destiny, we are waiting on you quickly.
Miss, she's not here.
All right. Let's have somebody else quickly.
Miss, can I ask a question?
Just a sec. Let me just have this one clear up and then you're going to
Miss-
ask any question. Yes, anybody else?
Miss, the blood. Miss, you could use the blood cell because it would stay up because oxygen is going in and oxygen is coming out at the same time. Oxygen is coming.
All right.
The blood,
So
the
look
blood
at this
cell,
here.
The red blood cell.
I want you to think about this here. An example of an isotonic situation is you donating blood to a person of the same blood type. Right? When you're giving blood to a person of the same blood type, because the concentration of your blood is the same as the concentration of that blood, the same molecules are present. So nothing will either come out of that person blood, nor nothing will go into your blood. No net movement will happen. So it is what you call an isotonic state. Okay? Everybody get that there? So when you have two two concentration of substance of the same concentration, there is no difference in movement, students. So if you have the same molecules, then there will be no net movement. That means to say it is therefore isotonic. Now, do you see why you need to have the same blood type and so on when you are looking at blood transfusion, so that there's nothing happening, but everything is blended together. And if there is a hypo or hypertonic, then the person might die. Okay, students? So that there brought us to the end of the session. We would have looked at osmosis. We reviewed the different types of cell content. We look at where osmosis occur. We use diagrams to show osmosis. We explain with the different cell contents, plants and animal cells, what can happen, and in reality, what or where isotonic, hypertonic, and hypotonic can be found. That there brought us to the end of this session on osmosis.